Thank you, Rodrigo, for the introduction. As Rodrigo introduced me, my name is Junior Nunes. I'm a PhD candidate at the University of Wisconsin-Madison. And I'm here today to talk about the implication of steroid cover crop adoption for amaranth management in soybean production. But before I go into the weeds, I just would like to start talking a little bit about myself, about my background. Okay. First issue of the day. So, yeah, there we go. As some of you know, I am from Brazil. I was born in the south part of the country in a small farm in the state of Paraná. I lived there until when I was about 15 years old. And then in 2010, my parents and I, we moved to the state of Mato Grosso. And that's where I started my career in ag. So back in 2016, I received my bachelor's in agronomy in the state of Mato Grosso. And then a couple of years before I finished my bachelor's, I started working with product development as an undergrad research assistant. And that was kind of my first contact with research, product development. I fell in love with research. So I transitioned from my undergrad to work as an independent researcher. I had of my own research company in Brazil. And then after a couple of years working, I decided it was time to go back to school. So back in 2019, that's when I started my master's degree in crop protection. So once a month, I used to travel to a neighboring state, the state of Goiás, to pursue my master's degree. And I received my master's degree in 2020. And like Rodrigo said, that was when I met Rodrigo back in 2019. He offered me the chance to come to Wisconsin for the first time back in 2020 as a visiting scholar. So it was my first year here in Wisconsin. And then in the middle of that program, Rodrigo offered me the chance to become a full-time PhD student. So in 2021, I returned to the US and started my PhD, and that's why I'm here today. So that's just a short bio. Now let's talk about what matters today, which is amaranth management in soybean production systems. So for those who don't know, waterhemp and palmeri amaranth, these are the two most troublesome weed species in soybean production regions across the United States. There are several factors that make those two weed species problematic. Some of those factors are related to their biology, but lately, one of the main challenges when it comes to managing amaranth species, it has been herbicide resistance. So farmers are concerned about herbicide resistance because they're losing options in terms of herbicides to control those amaranth species. So this is just a big picture of palmeri amaranth and waterhemp herbicide resistance over time. For both species, they had the first case reported back in the 90s. And then from there has been pretty much a linear increase in the number of cases every year. So those are some of the factors that make those two weed species so problematic. But they also have some weakness. So for instance, the fact that they are small-seeded broadleaves. So they have small seeds, which can give orange to small seedlings. And at those early stages of that plant, of those two weed species, those early stages, that's when those two weed species are more susceptible to management practice, such as cover cropping and pre-emerge herbicides, which will be the main topic of my presentation. So, as I said, we've been trying to use cover crops as a non-chemical approach to help managing amaranth species. I'm just gonna give you like a brief introduction how we can introduce cereal cover crop to a corn-soybean rotation. A corn-soybean rotation is a common cropping system in the US. Where we have corn in the first year and then soybean the second year. The challenge with this cropping system is that we have this fallow period between the two growing seasons where the soil is not protected. We don't have an active crop growing, so the soil is not protected against erosion, and also weeds have all the space they need to develop during that fallow period. So the goal with the cover crop is to replace that fallow period with an active plant growing in between the two growing seasons, so it can protect the soil, prevent erosion, and also help managing weed species. So the cereal is planted after corn harvest in the fall. That can happen between mid-September if it's corn silage, or it can go all the way to mid-November, depending how early the farmer can harvest corn. And then following cereal planting, we have cereal termination. That's when we kill that cover crop, usually with glyphosate as a chemical approach, to establish the next cash crop, which is soybean in this example. Just some basic information about the cereal, also known as winter rye. This is one of the most adopted cover crops in the US. And that's because cereal, first of all, can survive the harsh winter conditions that we have in the upper Midwest. And also it has a fairly fast growth rate in the spring, especially compared to other cover crop species. Cover crop is not something new, but it's something that has been gaining popularity among farmers. If we take the state of Wisconsin as an example, over the last 5 years or so, there has been about 20% increase in the adoption of cover crops, which represents about 12% of the corn and soybean area in the state. There are several reasons why we adopt cover crops. If you're a soil scientist like Dr. Huar, you're looking at soil erosion, controlling soil erosion, increase organic matter, reducing nitrogen leaching. But we as weed scientists, we use cover crops because cover crops provide weed suppression. And then when it comes to weed suppression, it all comes down to biomass production, growth of that cover crop. The more biomass we can produce that cover crop, the better weed suppression we can expect out of that system. But then in the upper Midwest, it can be quite challenging to grow and accumulate high levels of biomass, of cereal biomass, due to the challenging conditions that we have for cereal growth. We have a limited window for cover crop growth, not in terms of time, in terms of months, but in terms of temperature for growing degree accumulation and growth of that cover crop. So this is one of the main aspects of my research. I've been trying to understand how we can optimize the window for cover crop growth and biomass production. Another aspect of my research is the use of pre-emerged herbicides. Back in the 90s, in the past, farmers used to use a lot of trees, but then with the introduction of the Roundup Red technology, there was a decrease in the use of freeze, and now with all the issues that we've been having with herbicide resistance, especially to post-emerged herbicides, there has been again an increase in pre-adoption, especially in the corn, in the soybean market. But then when it comes to adopting cover crops, cereal cover crop, alongside with pre-emerged herbicides, there are some concerns, especially about the fate of that pre-emerged herbicide. So when we spray a pre-emerged herbicide, our goal is for that product to leave the spray boom and go to the soil solution to provide residual weed control. But when we have the cover crop, that biomass, the same biomass that can provide weed suppression, can also intercept that pre during application and challenge its ability to reach the soil and the soil solution. So understanding those interactions between cereal cover crop and pre-emerged herbicide is also another important aspect of my research project. So just to give you a big picture, the rationale behind my research projects, our goal with my project is to move away from what we call a conventional weed management, to be as close as we can to an integrated weed management. So in this conventional weed management, farmers rely a lot on tillage as a way to prepare the soil before cash crop planting and also provide a degree of weed control. And they also rely a lot on post-herbicide applications. So our goal is to make some modifications from that system. So for instance, replacing tillage with no-till, no soil disturbance, plus the cereal cover crop, protect the soil, prevent erosion, and also provide weed suppression. Also use an effective pre-herbicide program at soybean planting. As you can see, we're trying to be heavy early on in the season because that's when those amaranth species are going to be more susceptible to those management practice. And the goal of using the cereal cover crop in the pre-herbicide is to alleviate the pressure that we impose on post-herbicide applications, because most of the resistance that we have has been on post-herbicides. So there's more into integrated weed management than this, but we try to be as close as we can to this practice. So now I'm going to introduce you to my research projects. I have 4 total research projects, 4 chapters. And chapter 1, we try to elucidate the waterhemp suppression from the cereal cover crop biomass, understand how much biomass we actually need to have effective waterhemp amaranthus suppression, and also the mechanism behind that weed suppression. Chapter 2, we focus on pre-herbicide fate deposition. Unfortunately, I won't have time to cover chapter 2 entirely, but that's because chapter 3 is kind of an improvement of chapter 2, where we kept working with pre-herbicide fate in the soil and also evaluate residual weed control as an additional component. So And then chapter 4, it's the whole picture, the whole system in one chapter, where we evaluated cereal cover crop biomass production, amaranthus control, and also soybean yield. So understanding all the aspects of the system in chapter 4. So from now on, I'm going to go through chapters 1, 3, and 4. I'm going to briefly explain how we did the research and also the main findings, and then we move to future research right after. So starting with chapter 1, our goal here was to understand how much biomass we actually need to have effective water hemp suppression. So when we start working with cover crops, Rodrigo always saying like, we need biomass, but how much we actually need, how much is enough to have effective weed suppression. And so our goal was first to determine the number that we need, like the level of biomass that we need, and also understand the mechanisms of suppression behind that cover crop biomass. So the way we designed this study was to have as a dose response of biomass levels going from 0 all the way to 12 megagrams or 12 tons of biomass per hectare. And this study we only did in Wisconsin, in 2 locations, Wisconsin, Janesville and Brooklyn. So that's why I'll be talking about only water hemp in this project. But later on, you'll see I have multi-state efforts where we work with palmer amaranth and also water hemp. So to estimate, to simulate those levels of biomass with good precision, we start by harvesting the cereal from the field at antithesis. We then dry that biomass to constant weight, and then we weight those samples to meet those doses of biomass. Then we prepare the soil, and then we spread that biomass over the field to simulate the mulch effect from that cereal cover crop once it's terminated. So the goal here was to simulate what happens with the cereal cover crop mulch after termination. And then once we establish the study, we start collecting data. We did this study for a total of 5 site years in 2 locations in the state of Wisconsin. We collect some water hemp demographic data at 42 days after study establishment, like plant height, density, and biomass. My focus today will be on water hemp density. We also measure some soil abiotic parameters that would help us understand the mechanisms of suppression behind that cover crop. So we measured light interception by the cover crop mulch, and also soil temperature and soil moisture in the soil underneath that cover crop mulch. For all my projects, I used R and RStudio to analyze the data for all the steps of data analysis. I'm not going to go into details from the data analysis because it takes a lot of time to explain that part. So if you have any questions about data analysis, coding, models, whatever question you might have, please feel free to ask at the end or even after the presentation. You have my contact. So just as an example, for this project, since we have those response curves, we use nonlinear regression models from the DRC package to analyze the data. So that was based in R. So moving to the main findings of this research project, starting with water hemp density at 42 days after study establishment. So in each one of those boxes, we have the 2 locations that the study was conducted, Brooklyn and Janesville. On the x-axis, we have the cereal biomass in megagrams per hectare. And the y-axis, we have water hemp density. And each one of the lines represent one of the years that the study was conducted. So when we look at this plot, just like I said in my introduction, the more biomass we can have in this system, the better weed suppression we can expect out of the cover crop. So in some site years, when we have about 12 megagrams of biomass, 12 tons of biomass, we had pretty much 100% reduction in water hemp density. There's no herbicide in this study. This is just a cover crop effect, just a mulch effect from that cereal. So, and with this study, we also managed to estimate that we need about 5.2 megagrams of biomass, 5 tons of biomass to reduce water hemp density by 50% compared to bare ground conditions, compared to the check, no biomass. And biomass in this study is something that is so important because if you have enough biomass, the correct amount of biomass, you can expect suppression out of the cover crop. On the flip side, if you have low levels of biomass, in our study below 2.4 megagrams of biomass, we had the opposite effect. When we look at those plots, at low levels of biomass, we can see there's always an increase in water hemp density compared to the check. That's because under low levels of biomass, that mulch can hold moisture underneath the soil, in the soil underneath that mulch, and that moisture can stimulate water hemp germination. So that's why we measure soil moisture in this study. And as you can see for all site years, as we increase biomass, there's also an increase in soil moisture. And besides affecting soil moisture, the cereal cover crop biomass also had a tremendous impact on light interception. So what happens with this system is we have the cover crop mulch protecting the soil, the sunlight is gonna reach that mulch, and the mulch is gonna block the sunlight, it's not gonna go through. So the effect of that light not going through that mulch is that the mean soil temperature underneath that mulch is a lot lower compared to a bare ground condition. So we measure soil temperature in this study. So this is an overall picture of soil temperature. So we have the hours of the day going from midnight all the way to 11 p.m. And then each one of those lines represent a level of biomass. So when we didn't have biomass, the red line, just bare ground, look at the fluctuation soil temperature throughout the day. It went from 19 Celsius all the way to 28 Celsius when we didn't have biomass. And then as we start adding biomass, 4.8 and 12 milligrams, that temperature fluctuation reduces a lot. And the reason why I'm focusing so much on soil temperature from this study is because when it comes to waterhemp germination, temperature is the main driver. So I took this data out of a paper from the literature where they evaluated waterhemp germination at different levels of temperature. So they have levels of temperature going from 5 all the way to 35 Celsius. And they have two types of temperature in this study. One, the temperature was kept constant the whole time. There's no change, it was just the overall average here. And on the flip side, they also allowed that temperature to vary 40% above or below that average. So when we focus on this data, we can see that as we increase temperature, overall temperature, there's a response in waterhemp germination. And when we focus on the 25 Celsius average, when the temperature was constant, there's no change in temperature, it was 25 Celsius all the time, waterhemp germination was about 10%. And when they allow the temperature to vary between 15 and 35 Celsius, waterhemp germination was almost 80%. So that's why temperature is so important in this study, because waterhemp needs that temperature fluctuation to have good emergence. So that brings me to my main conclusion from this chapter. First of all, the cereal biomass can effectively suppress waterhemp, and the level of suppress is going to respond to the level of biomass. More biomass, better weed suppression. On the flip side, low levels of biomass can have the opposite effect, can stimulate waterhemp germination because it can hold moisture underneath that mulch. And finally, we believe that waterhemp suppression is due to the light interception from the cover crop mulch, which lowers the soil temperature and temperature fluctuation. Since waterhemp needs that temperature fluctuation to germinate, we believe that is the main mechanism of suppression from that cover crop. And one of the main goals of this study was to tell farmers when do they need to terminate that cover crop, because this was a common question when we start working with cover crops. Farmers were always asking, when do I terminate my cover crop? How much biomass do I need to have effective waterhemp suppression? So by estimating that we need about 5 micrograms per hectare of biomass, we can use that information to provide some guidance to farmers when they should terminate that CRI. Because CRI termination is one of the main aspects when it comes to biomass production. The more you can let the CRI grow, the more biomass it can produce. So there are two major recommendations for terminating the CRI. One is the early termination. So that's when farmers terminate their rye about 2 weeks before soybean planting. The problem with this recommendation is that, especially in the upper Midwest, most of the time, depending how early the farmer terminates that triggers that early termination, there might not be enough biomass to provide effective weed suppression. So we've been trying to adopt a new practice, which we call the planting green system, where farmers delay termination until soybean planting. So they delay the termination about 2 weeks for about 2 weeks. So we can extend the window for cover crop growth and biomass production. So we have a whole multi-state effort with several other states just to compare those two termination methods. So this is a really important research project for me because I took 2 chapters out of that project, chapters 3 and 4. So we conduct this multi-state effort back in 2021 and 2022 for a total of 19 site years of data. And then we evaluate several aspects of the system from this project. So for chapter 3 only, we selected 4 locations: Kansas, Illinois, Wisconsin, and Pennsylvania, just to focus on herbicide fate in the soil. So we select those 4 locations to focus on chapter 3, where we compare the early termination of that CRI versus planting green in terms of herbicide fate in the soil and also residual amaranthus control. I'm going to refer to Amaranthus from now on, because with this multi-state effort, we work with waterhemp in Wisconsin, but people in other states, they work with palmeri amaranth. So that's why I'm going to refer to as amaranth instead of just waterhemp. So our goal here was to compare herbicide fate and residual weed control. And I'm going to explain now the whole set of treatments that were evaluated in this project. And then I'm going to tell you which ones we selected to work with herbicide fate just for chapter 3. So in this study, we have a 2x3x2 factorial, so 12 treatments in total. The first factor we evaluated was soybean planting time. So we had 2 soybean planting times, early and late. The early was about 2 weeks before. The early was the earliest stage for planting soybeans in each one of those locations based on their conditions. And then the late was about 2 weeks after the early planting time. The second factor was the cover crop management. So we had no-till, no cover crop. And then the CRI treatments were divided into early termination and planting green. The early termination was about 2 weeks before each soybean planting time, and the planting green was at soybean planting for each one of those planting times. And for both terminations, glyphosate was used as a chemical termination of the CRI. And the final factor was the use of a pre-herbicide program. So half of treatments received a pre-application at planting, and half of the treatments did not receive a pre-herbicide application at soybean planting. And then for chapter 3 only, we selected the no-till, the early termination, the planting green out of only the late soybean planting time to work with herbicide fate. So again, we selected no-till, early termination, and planting green out of the late soybean planting time to measure herbicide fate. So we collect soil samples at 0, 7, and 21 days after treatment, after pre-herbicide application, to quantify flumioxazine and peroxyphone in the soil over time. So moving to the main findings of this research project, here I have an overall summary of peroxyphone and flumioxazine concentration in the soil over time. This is an average across site years. So in each one of the boxes, we have one of the herbicides. On the x-axis, we have our days after treatment when we soil sampled. And then on the y-axis, we have herbicide concentration in the soil. And each one of the lines represent one of the management practice. So the red line is the no-till, the blue line is the cover crop early terminated, and the green line is the planting green system. So when we look at this plot, one of the main trends we can see is that as time progresses, there is a reduction in herbicide concentration in the soil over time, compared to 0 days after treatment for most of those treatments. And that is because once we spray a pre-emerge herbicide in the soil, there are dissipation process that can happen, and that can dissipate the herbicide concentration from the soil. But then when we compare the different management practice, we can see that no-till always had a higher concentration for both herbicides compared to early termination and planting green. But when we compare the 2 termination timings of that CRI cover crop, there's not a lot of difference between the 2. If we go to 21 days after treatment, they're quite similar in terms of concentration of those 2 herbicides in the soil. So our best conclusion from this study is that, when you delay termination, you can have more biomass so that you can have more interception. But there's not a lot of difference between spraying pre's over dead versus green biomass based on our findings. And when we compare the difference between no-till and cover crop treatments, which was the main trend here, we can see that the largest gap happened at 0 days after treatment. And that is because we have pre-herbicide interception at the moment that we spray that product over the cover crop biomass. So this, and then when we look at the effect of cover crop biomass production on pre-herbicide concentration in the soil at 0 days after treatment, we can see that for both herbicides, as we increase biomass, we increase ground coverage, there's a lower herbicide concentration in the soil for both herbicides. And this was also focus of chapter 2. I won't have time to cover chapter 2 entirely, but just want to, touch a little bit on. So for chapter 2, we also work with herbicide fate in the soil, and we also measure pre-herbicide deposition. So the spray deposition using spray cards, water-sensitive cards underneath that cover crop mulch. So this is just 2 examples of 2 cards from tillage and no-till in terms of spray coverage. As you can see, there's not a lot of difference between tillage and no-till. But when we look at the CRI planting green treatment, on average across 3 years, there was about 70% reduction in spray deposition compared to tillage. So that biomass can intercept those spray droplets during pre-application, and that's why we observe a lower herbicide concentration in the soil compared to tillage and no-till. I want to use this illustration just to simplify what we learn based on our studies and also based on information from the literature in terms of herbicide fate in the soil when we have cover crops. So let's say we have a standing stand of CRI and we just spray a pre-emerge herbicide in this system. So the first thing that can happen during pre-application, we can lose herbicide due to drift. That's something that can happen. And then from the herbicide, from the proportion of the herbicide drops that were not affected by drift, we can have herbicide that can go straight to the soil, will not be intercepted by that biomass. It can travel between the CRI rows, the CRI biomass, and can go to the soil during application. But part of that herbicide can be intercepted by the biomass, by that CRI cover crop. And then from the proportion of the herbicide that was intercepted by that cover crop, part can move to the soil with rainfall, falling application. That's why it's important to have rainfall, falling application. The sooner, the better. But part of that herbicide can dissipate to the environment due to several factors. And part of that herbicide can remain adsorbed to that biomass and never move to the soil. No matter how much rainfall you have after application, it will not move from that biomass. And what we change from situation to situation is the more biomass there is at pre-application, the more ground coverage, the more interception. The higher is going to be the interception, and the lower is going to be the concentration of the herbicide in the soil at application. So the more is going to move after with rainfall following application. So moving to the main conclusions out of this chapter. First of all, the CRI biomass, the CRI treatments reduce flumioxazine and peroxyphone concentration in the soil compared to no-till at all sampling times. And delaying CRI termination, since there was more biomass at the planting green system compared to the early termination, we observed a little bit more interception from that pre-herbicide, and that's why there was a lower herbicide concentration in the planting green compared to early termination. There's not a lot of difference between the 2 termination methods. And finally, I did not show data for this, but I will show data for this conclusion, chapter 4. Regardless of the herbicide interception, lower concentration in the soil, we never observed any negative impact in residual amaranth control from the CRI cover crop treatments. So this is good news to farmers because whenever we present this data, farmers, they always ask, so if I have a lower herbicide concentration in the soil, does that mean I'm going to have a lower residual amaranth control? And based on our findings, the answer is no. So there's no negative impact when you combine cover crops with pre-emerge herbicides. Now moving to chapter 4, this is the whole system in one single chapter. So again, this is part of the multi-state effort conducted over the 2021 and 2022 growing seasons. We have a total of 19 site years of data. And the overall goal of this chapter was to compare the difference between early termination of that CRI versus planting green in terms of biomass, CRI biomass production, amaranthus residual control, and also soybean yield, stand and yield at the end of the season. So this is the whole set of treatments that I just explained. We had 2 soybean planting times, pre-cover crop management practice, and the use and no use of a pre-herbicide program at soybean planting. And the focus here, again, was on biomass production, CRI biomass production, amaranthus control, residual control, amaranthus because we had palmer amaranth and water hemp in this project, and soybean stand and yield at the end of the season. So moving to the main results, starting with CRI biomass at the time of CRI termination. Here I have So here I have biomass production in terms of termination timing and soybean planting time. So the two termination times we have, the early termination planting grain, and the two soybean planting times, early soy and late soy, and this is an overall average across site years. And then we have biomass megagrams per hectare on the y-axis. So when we delay cereal termination, either by delaying the termination itself or by delaying the soybean planting time, we observe between 30 and 40% increase in biomass production. And then if we focus on the effect of the cereal termination timing with the planting green system, we observe an average of 6.3 megagrams per hectare of biomass. Chapter 1, I said that we need about 5.2 tons of biomass to have about 50% reduction in water hemp density. So if you take that threshold of 5.2 as kind of a measure of success of this system, we can say that overall, across site years, planting green was a successful recommendation to go beyond that threshold. However, out of the 19 site years of data, in 8 site years, we did not observe an overall average of biomass production beyond that threshold. So for those site years that did not observe an average beyond that 5.2 megagrams of biomass, there are several factors that impacted those site years. Some of them was the cereal planting date. Some site years planted the cereal after November 5th, which was a little bit too late to have an early start of that cereal before winter. Also, limited precipitation between cereal planting and termination. That was also a limiting factor. And finally, also, they had some issues with cereal establishment because of high amounts of corn residue. So this is something that we always observe in Wisconsin as well. When we have too much corn residue from the previous crop, the cereal doesn't grow as fast as compared to a no-till corn soybean stubble. And this information, when we take this information out of this research project, it's important because we take that information, translate that into the management decisions to farmers, so they can use that information so they can have an optimum condition to grow the cereal cover crop. So now moving to Amaranthus density at the time of post-herbicide application. So we have, again, palmer and water hemp in this data set. So here in the overall average of Amaranthus density in plants per square meter. So each one of the sides of the plot, we have no pre and yes pre. And then on the x-axis, we have our different management practice. So we have no-till, early termination of the rye, and then also the planting green system. And on the x-axis, the y-axis, we have Amaranthus density. So if we focus on the no-pre side of the plot, we can see that the no-till had the highest Amaranthus density overall, and the planting green had the lowest Amaranthus density. Over site years, we observe an average of 53% reduction in Amaranthus density compared to no-till. So in this study, we had an overall average of 6 megagrams per hectare of biomass, and we had 53% of Amaranthus production. And in chapter 1, we had like 5. We estimated we need 5 to have 50% reduction. So the numbers are quite close. And then when we focus on this yes-pre side of the plot, there are smaller difference across management practice. And overall, when we combine the planting green system with the pre, we observe about 80% reduction in Amaranthus density compared to no-till, no-pre. So this is the data to support my conclusion from chapter 3, where I said that we didn't observe any negative impact on the residual Amaranthus control when we combine the planting green with the pre. No matter how much herbicide interception was in that treatment, there is no negative impact in terms of residual Amaranthus control. Now moving to soybean yield and stand at the end of the season. So starting with yield, each side of the box, we have the 2 planting times, early and late. On the x-axis, we have our management practice, no-till, early termination planting green, and then soybean yield on the y-axis. This is an overall picture across 19 site years of data. And when we summarize the data, we observe that the planting green system reduced soybean yield from 8% all the way to 18% compared to each one, compared to no-till in each one of those 2 soybean planting times. So this is concerning because so far we saw that planting green was beneficial in terms of biomass production, Amaranthus suppression, but now soybean yield is not looking so good, so promising. So we decided to look why that yield reduction was happening. So that's why we collect soybean stand count at the end of the season. And this overall average is a good representation of what we observe from the soybean yield data. The planting green system reduced soybean stand between 11 all the way to 17% compared to no-till in each one of those two soybean planting times. So at this point of my program, I had finalized summarizing 19 site years of data. I had a large data set in my hands. I knew that the planting green was reducing soybean yield, and soybean yield also had, there was a degree of correlation between stand and yield. So the question at this point was, what do I do now? So how can I prove that stand is affecting soybean yield? And what other factors can also account for that yield reduction? Is there any other factor that can account for yield reduction besides soybean stand? So at this point, this is when we ask for help. So I just want to open like a parenthesis in the middle of the presentation, share an experience that I had through my program, which was to work with Dr. John Wallace at Penn State. He's a wheat scientist at Penn State, and he is also part of this multi-state effort. So when we finalized summarizing the data, Rodrigo and I, we met with him and we asked his opinion. What should we do in terms of data analysis to explain this relationship between stand and yield? And he suggested us to use a structural equation modeling, also known as SEM, to provide like a good representation of the entire data set. So I had the chance to go to Penn State last fall. I spent a week working with Dr. John Wallace, and it was a fantastic experience. So I just want to use this opportunity to thank him for all his effort with the project. So when we work with SEM, the number one rule is to have a really large whiteboard so we can lay down all your hypotheses. You're going to spend a whole morning going back and forth on those hypotheses before you go to R. I will not explain the stats, the math behind SEM, the coding. If you have questions, please ask. But to give you like a brief explanation what SEM is, let's say you have two predictors, X1 and X2, and then those two predictors can help explain a response variable Y1. So far, this is nothing new. This is linear regression. But then the neat thing about SEM is that we can use that Y1 response variable as another predictor, can also act as a predictor to help us explain a second response variable. So this SEM, this is why they call path analysis, because you can have a large data set and then you can explain the relationship between predictors and response variables in your data set. So we did an SEM model for soybean yield, and this is the overall picture of soybean yield. There's a lot going on in this picture, so allow me to walk you through. The easiest way of reading this picture is to go from top to bottom. So on top, we have the planting green C-right termination timing and the late soybean planting timing. And both of those predictors had a positive relationship with biomass production. The size of that effect is measured by the standardized coefficient. So 0.29 for planting green and 0.32 for planting time. So this is just telling us that as we delay C-right termination, we're increasing biomass production. This is what we saw a few slides back. But then biomass production had a negative relationship with soybean stand. 0.3 was the size of that effect. And then soybean stand, on the other hand, had a positive relationship with soybean yield. So higher stand, higher yield, higher population. But as you can see in this picture, in this image, there's no relationship, there's no direct effect of biomass on soybean yield. For biomass, for C-right biomass to impact soybean yield, it has to go through soybean stand. So soybean stand acts like a mediator in this study. So a way we can look at this is like a decision tree. So let's say we have soybean established, soybean stand on the top. And then if that soybean established is successful, there's a low chance of having any kind of yield penalty when planting green. But then on the flip side, if soybean establishment is unsuccessful because of that biomass, there's a high chance of having some impact on soybean yield at the end of the season when planting green. And then this is just to explain some of those relationships, some of those effects. So for example, the effect of biomass on soybean stand. For some site years, there was a negative relationship between the two. And then most of those site years, we also observe the positive relationship between stand and yield. So this is how we explain, this is how we took all those findings. So moving to the main conclusions out of this chapter. First of all, the planting green recommendation was an effective way of optimizing biomass production, CRI biomass production. And by increasing biomass production, we also had a benefit in terms of amaranthus suppression. Just with the biomass, the overall average was 50% reduction in amaranthus density across site years. Adopting an effective pre-herbicide program was also important in this study. When we combine the planting green recommendation with the pre-herbicide program, that was when we observed the highest reduction in amaranthus density overall. However, the planting green had an overall reduction in soybean yield compared to no-till in both planting times. And that yield reduction was because some negative impact of biomass production on soybean establishment in some site years. So this summarizes chapter 4, all my chapters. And now just to give you a conclusion from all my research chapters after a couple of years working with cover crops, I conclude that the main piece of the puzzle here is biomass production. Biomass can impact everything in this system. So for instance, biomass can impact amaranthus suppression. More biomass, better weed suppression. Biomass can also impact pre-herbicide fate in the soil, can intercept pre during application and reduce pre-herbicide concentration in the soil over time. CRI management, so biomass will not impact management of the CRI, but if you want to be proactive and accumulate the desired level of biomass that you need to have effective weed suppression, you need to optimize your management, optimize planting time and termination timing of that cover crop. And finally, soybean management can also impact biomass production, depending how early or late the farmers terminate and plant the next cash crop, and dictate how much biomass that CRI can produce. And biomass accumulation, the flip side, can also impact yield if there is any kind of issues with soybean establishment when planting green. And then based on our findings from those research projects, I think there's a lot of room for new research. And one of the main questions that I have now is what happens with the amaranthus seed bank underneath that cover crop mulch when we have weed suppression. So in this picture, we can see there's the cover crop mulch. There's no amaranthus plants growing in between that mulch. But when we look all around the plot, all we can see is amaranthus plants. So the question is, what happens with those seeds underneath that mulch? So one hypothesis that can explain this is that the mulch effect, the effect of that mulch on soil temperature is so large that there's no seed germination. So seeds are not even germinating in the first place. And then the second hypothesis that the seeds can germinate, but since amaranthus species, they have small seeds, they have a limited amount of resources to go through that residue, so they can fail to emerge, they can die on the way. Those two hypotheses explain why we see suppression, but the issue with if hypothesis number one is true, those seeds can be a problem for next year because they can remain viable for a next growing season. Another aspect that I think needs more investigation is also in terms of pre-herbicide fate in the soil when we have cover crops, especially in the planting green system. So here I have the main pre-herbicides that are labeled for soybean production systems. And I think it's valid to trying to understand which of those AIs, single AIs, are more compatible with the planting green system in terms of rainfall accumulation to move that herbicide from the biomass once it's intercepted all the way to the soil, following application, and also residual weed control. So I think those two aspects can be further evaluated. In our planting green project, we had a lot of issues with soybean establishment, especially under the high levels of biomass accumulation. So we need to find ways of going around those issues with soybean establishment. We already have one ongoing research project investigating this, trying to go around that recommendation, those issues. And the way we found to go around this, the way we're evaluating this is instead of waiting for the CRI to accumulate that threshold of biomass that we need for effective weed suppression and then killing that CRI in the same day that we plant the beans, we've been trying to plant the soybeans very early in the season when the biomass is still low, so we don't have any kind of issues with soybean establishment, and then kill that cover crop later, a couple of weeks later, let the rye grow, and then we can kill later when we have that threshold of biomass production. So with this study, we managed to reduce the impacts of the CRI on soybean establishment because there is low amount of biomass at soybean planting, so there's low chance of issues with establishment. And we also been able to measure how long that soybean can withstand competition with the rye following soybean planting. And finally, I think there's a lot of agronomics that deserve more attention in terms of planting green. So for instance, soybean genetics, I believe there are some varieties that are more compatible with the planting green system. Perhaps varieties that can branch a little bit more, can compensate for potential stand losses. Soybean population, I think we need to understand what is the minimum soybean population that we need to have to maintain good yield potential when planting green. Does that change from the recommendation that we have from no-till and tillage? Row spacing is also another agronomic. Reducing row space, we have a lot of benefits in terms of weed suppression, weed control. So does that impact, how much population we need to have maintain good yield potential? And planter adjustments, as we saw, there are some cases where we have some issues with soil establishment due to a high amount of biomass. So farmers need to understand that when they move from no-till or tillage, they need to make some changes in their planter setup, to plant through a high amount of biomass. So we need that information so we can guide farmers for the best recommendations. And also some aspects of fertility management. We know that the CRI can impact nitrogen availability early in the season. So that's why planting corn green doesn't work most of the time. But what about soybean? Is there any other limitation in terms of fertility management? So that concludes my presentation. I just want to take a few moments to thank all those involved in this journey.